Hey, welcome everybody to the High Performance Computing Symposium for fall 2024. I think we're sharing a quick agenda on here, so I'll just kind of give a welcome and a brief introduction of some of the cyber infrastructure HPC resources, a little history, and then we will roll into the wonderful lightning talks that folks have planned. To highlight some of the research that's been going on. So, yeah, I'm Sean Cleveland. I'm the acting director of cyber infrastructure, and I've been for about the last 10 months. So again, welcome. Um, so I'm just going to briefly hit kind of our road from Mana to Koa here, how UH kind of got some HPC resources, a little bit about the Hawaii Data Science Institute, some other services that we have, and then we'll kind of roll into lightning talks. So a little history. University of Hawaii as a system didn't have a system high performance computing cluster that was centralized and available to everybody for free until about 2014. So since that time, we've gone from 184 nodes to almost 300 nodes with the present Koa cluster. We had no GPUs. Now we have almost 150, and we've more than doubled the core count on this. And so we went from 7 users now to almost 1,400 folks that have gotten user accounts on the cluster, and about within a typical year, we're up to about 460 folks that are active. We had a $1.8 million initial investment, and through that time, we've had additional investments through PI folks buying in condo nodes at almost $1.6 million, and additional $1.6 million in grants. So we're at about $5.2 million in investment of high performance computing infrastructure at UH within the last about 10 years. So a couple highlights in the last year. We were up 12% for users on the HPC system compared to the previous 2023 year. 15% more active users than the prior year. We saw an uptick in folks using CPU hours, a giant kind of leap in GPU usage in the last year. So that's great. We have those resources, and we're excited that folks are leveraging those. The wait time to run jobs went down, and then one of the other highlights that I mentioned in a previous meeting that we're excited about is folks that are, getting publications out of some of the data and jobs that they're running on the HPC and acknowledging our resources. So we're excited about that. We had 36 papers acknowledge Mana or Koa last year. So what kind of goes hand in hand with some of the HPC resources is the Hawaii Data Science Institute. So cyber infrastructure is a part of the Hawaii Data Science Institute. If you're not familiar with what this is, it is a UH system-wide initiative to catalyze research and training capacity within data science, computation, visualization tools aimed at students, researchers, as well as looking to forge industry partnerships. So the Institute was established in 2018, so almost 6 years now that it's been around. A couple highlights from last year, since the beginning. The Institute's given about 44 different workshops with almost 1,500 folks that have attended those, and about 38 seminars with almost 850 folks in there. We also have some student summer programs that we've had in the past for undergraduate students to participate and learn data science skills in sort of an immersion over a week. And so there's just some stats and breakdowns of number of PhD, masters, and sort of bachelor students that have also been involved in our fellows program, and are also some of our folks that are alumni from the Data Science Institute. So we do have a fellows program that graduate students can participate in. There's a new one here, opportunity for HIDSI Junior Data Science opportunity for students and graduate students that I wanted to kind of mention here. So You can look more into this, but students that are accepted, you need to attend some of the workshops that I kind of mentioned in the fall and the spring. There's access to AI micro-credentials, self-paced data camp modules, and things like that. So this is kind of a new kind of fun opportunity for folks to learn more about AI and get some AI skills. Other highlights from some of the students that are part of the Data Science Institute. We had a grand prize winner in visualization from the AGU 2023. RJ Tabalba was one of the award winners there. He's in Jason Lee's lab in the Lava Lab next door. Super exciting there. Two Nature papers from some of the HIDSI alum, and then an additional couple of manuscripts that are in preparation. So we're very excited about those. Additional services, just to mention here, so we have HPC, we have storage. We also offer software engineering and data science services as part of cyber infrastructure and the Hawaii Data Science Institute. So we partner with Department of Health. We help with the Hawaii Climate Change Data Portal, doing data science and software engineering things. So if you're a PI or a researcher and you need some of these services, let us know. We're happy to help. One last resource I'll talk about is Jetstream 2. This is an NSF cloud resource that there is a piece of this infrastructure at the University of Hawaii. So it allows you to spin up virtual machine servers. It has access to A100 GPU accelerators, both at UH and then at the larger Indiana University infrastructure there. So if you're needing to run long-running jobs, web services, databases, you need some dedicated computing resources, Jetstream 2 could be an option for you. So feel free to reach out to us and ask us about this so we can help you get sort of a starter account on NSF access to use Jetstream 2. So with that, that's kind of the welcome that I wanted to get into and leave time so that we can get into these great lightning talks. So I think our first speaker is Kacie Kajihara. She's a PhD student at the Hawaii Institute for Marine Biology, and she's going to talk about network-derived indicators of microbiome stability across a Hawaiian watershed. So I will stop sharing.
Am I supposed to like unmute, or is there like how to?
No,
it's working. I can hear you.
Okay, thank you. Okay, so hi everyone. Yes, my name is Kacie. I'll be presenting on my master's research, which recently completed using networks to look at microbiome stability across the entirety of a Hawaiian watershed. And so, in essence, this was a project about microbiomes. For those of you who may not be aware, the microbiome is just the collection of microorganisms like fungi and bacteria that I looked at here, as well as things like protists, viruses, and archaea that inhabit a given habitat or organism. They play huge roles in maintaining the health and function of all life on Earth, whether that's on the individual host level, like in our own human gut microbiomes, or in maintaining global biogeochemical processes. And so the health of the ecosystem really does hinge in large part on the microbiome and its ability to respond to disturbance. And that's where our interest in microbiome stability really lies. Microbiomes are pretty complex, though. They're made of many different organisms that are interacting with one another in pretty complex ways. They could be helpful, harmful, neutral. They're oftentimes interacting with many different things at once, not just one. And so one cool tool that we can use to model an interaction, or like the context of these interactions, is a co-occurrence network. And so every single node in the network corresponds to a microbial taxon, and then we draw an edge between them when we think that there's a significant relationship between them. And the density of these edges and other properties of the network can be measured and used to describe the stability of the overall network. So we're using that to model the stability of the microbiome. So the general premise of the project was to see if using these networks and their properties, do I find that there are any universal network properties of microbiome stability across a diverse range of habitats and environments? That range of environments came from the Waimea Ahupua'a, which is located on the north shore of Oahu. So there was a really big microbiome census done a few years back where they looked at all the fungi and bacteria from mountain to ocean, different environmental conditions, different sample types within. And that data set is what I was able to use for this research. And so kind of around the edges of this slide, we see the different diversity of sample types that we had available. Many of the other studies, just in this particular field of like network microbiome stability, really only look at soil alone. So the fact that we had these 13 different sample types was very cool. That added up to about 1,400 total samples. The sequencing effort gave rise to several thousands more of fungal and bacterial taxa. And this is really where the bulk of my HPC usage came in, because when you're making a network, you're doing a lot of pairwise comparisons. It's not super readily parallelizable. And so even making a slow network can be pretty, or making a small network can be pretty slow. Making a network on this size would have been impossible without these resources. So when I'm looking at what makes for a universal property of stability, it's something that unifies the different spatial scales in the watershed. So I can have a network that looks at the whole watershed, the habitats that make it up, as well as the environmental gradient from mountain to ocean. And then that came out to a total of 33 networks when you're parsing these categories. I also have fungi, bacteria, and other like microbial kingdom combinations that end up making this total. And so this is really just to hit home that HPC made my thesis possible. Thank you very much for that. Even after generating your network, a lot of the metrics that we use do rely on pairwise iterative calculations. And so I didn't attempt anything on my desktop computer really. This is a lightning talk. So the SparkNotes version of my entire thesis is that 3 main universal properties kind of arose from this data, which were we found more stable or robust networks when they were interkingdom links, when there was a higher proportion of negative edges in the network, as well as more diversity amongst the bacteria. So for every single scale in that table that I showed you earlier about like space, I did generate fungal, bacterial, and interkingdom networks. Interkingdom just meaning there is fungi with bacteria. I did find consistently that when you have fungi with bacteria, we find more robust networks, which robustness was kind of like our metric to represent stability in this study. It basically just means in my entire network, I take nodes out one by one and I measure how fast that network starts to fall apart. These curves are kind of following the fate of that process. General rule of thumb is that you can look at the area under the curve, where like a lower area under the curve means lower stability. You can also see some pretty steep drop-offs in some of these curves. So basically the blue lines, which are the fungal networks, are losing out on like all accounts. I'm not showing you the gradient networks here, but the pattern across the watershed was that the interkingdom networks represented in yellow here were more stable than the single kingdom networks. And then another result was that the fungal networks, which I saw were the least robust, also had the smallest percentage of negative edges, whereas the interkingdom networks had the most. This supports the result that we found that fungal networks were really stable, interkingdom were most. Might be a little bit counterintuitive because they're negative interactions. That could be things like predation or parasitism. But when you're looking at like an entire ecosystem, we do expect to see more of those in a more stable assemblage. And then the last result is that when you're looking across kingdoms instead of across space, there are certain sites that pop out as consistently less robust. Here it was the marine habitat that's showing up in the light blue line here. One correlation with that site was that the bacteria there were more similar to one another, so they were less diverse. Another way of saying that is that the more stable sites we found to have higher levels of diversity, which makes sense because they can have more functional redundancy that could help in case of a disturbance. But one interesting result, or this result was pretty interesting just because the identity of these sites were ones that we would expect to contain more desirable ecosystem states. And so on the habitat side, we have a marine protected area. The less robust site from that environmental gradient was all the way at the top of the mountain. It really has like no human activity compared to some of the more like tourist attractions lower in the valley, like the waterfall that everyone swims in at Waimea Valley had a much more robust, stable network. So it just goes to show that stability isn't a universally positive thing. Like in nature, we can have invasions, disturbances can select for things that might not be the most beneficial and can also be very hard to get rid of. So it's important to consider that duality. And in conclusion, We find that under this broad umbrella of complexity is what appears to underpin stability. So when you have complexity in terms of like your edge type or what kinds of links you have in there, the network seems to be more stable, which may represent a more stable microbiome. And then it's important to consider that although stability has been thought to be like an important facet of microbiome health, it is indeed a facet and probably should be considered alongside other things if you're looking to like engineer a microbiome to restore a disturbed habitat. Yeah. Thank you for having me, and thanks to the HPC for the resources that made this work possible. This is also like a huge collaborative effort to sample like an entire watershed, and so there are many other thanks to go out there as well. But yeah, I'd be happy to take any questions.
I have a question for you. I'm on Zoom. Can you guys hear me?
Yes. Yes.
So when you were sampling, and did you do any like specific species ID for like fungal and bacterial species?
We left it at the level of operational taxonomic units, which it's kind of like it's arguable, but it doesn't get to species level resolution, no.
Okay, thank you.
Good. Were you using any particular software pipelines for any of your kind of analysis in here, or just some of the network?
Yes. Yeah. So Speakeasy was the network software that I used. I think it integrated with another package called Huge that enabled it to be like I could use the batching so that it would actually run even with the HPC help. And then iGraph was the main kind of like network metric measurement package.
How long did those runs take typically?
Okay, I do actually have one like right now that I think will finish in about like 5 days. And I'm probably like not using all of the resources that I'm requesting. Like I think it's like it doesn't take a huge amount of resources, but it just takes a long time and probably like the more cores, the better.
Great. Yeah, nice not to have to leave your laptop on for a week at a time.
Oh
And
yeah, I love that. I've made so many Slurm scripts.
okay.
Okay, thank you.
Yeah, thank you, Kacie. This was great. Okay. Let's see. Is it Komal Yadav? Is she present in
Maybe like a whole like Nicola, sorry. Yeah. Yeah, definitely. I had it in my head that I'm going second because the agenda said so, so I'm going. Spotify. Okay. Okay. All right, so I think it's also appropriate if I go because I want to continue the microbiome train that Kacie started. So I am jointly co-advised by Dr. Baderos and Dr. Hinson, and we are in the PBRC. And my postdoctoral research has mostly focused on mosquitoes, and specifically this early life stage that they have, where if you go pretty much any stagnant body of water around campus right now, you'll probably find this early life stage. They're very good at what they do, which is bad news for us since they are annoying, first of all, and second of all, they spread diseases everywhere. So one part of this early life cycle that's really important is the composition of the detritus. So what leaves have fallen in and are decomposing and releasing nutrients. So the early life stage of these mosquitoes are detritivores, so they consume detritus. But what's really understudied and what I'm going to be focusing on is what role are the microorganisms playing in this little micro food web in these little bodies of water. So the microbes could be helping the mosquito host directly by living inside them and doing things sort of like our gut microbiome. or they could be helping indirectly by helping break down that detritus into nutrients that the mosquito can use. So how do we do this type of research? Well, if you just take one tiny little drop of pond water from this stagnant body of water that the mosquitoes are in, you'll get an average of 10 to the 5 or 10 to the 7 cells per milliliter, so just chock full of microbes. You can't see them, so we turn to fancy sequencing technologies like Kacie was just showing. And this helps us determine around how much relative to each other, how much of our sequencing reads are coming back from which bacteria. And we use the 16S rRNA gene. And the average data set that I'm working on is usually around 15 gigabytes of sequencing data, which is crazy from just this tiny little drop of pond water. But I am very thankful to the HPC because this is the current memory storage on my local laptop that I'm presenting to you on. Very mismanaged there. Very grateful to the HPC also for the C-MyIki gateway, which I believe Kacie also used. So I can give them my raw sequencing data, which looks like what you see on the left with all the A's and G's and T's. So that's our DNA sequence. The 16S pipeline is a Nextflow pipeline that runs all this stuff. I can like close my computer and go away while it runs overnight. And this tells us what types of bacteria I might have in my data based on close taxonomic matches to the sequences. So I'm just going to jump into an example of how I've used this HPC resource into one of the experiments that was actually just recently accepted. So we took eggs from this mosquito, Aedes albopictus. We got rid of as much of the bacteria as we possibly could. There's a few exceptions, which we're not going to worry about today. And we distributed these early life stage of the mosquitoes called the larvae into a bunch of different treatments. So the first one that we did is a detrital infusion where we steeped native leaves, leaves from a native tree called Ohia lehua, steeped them for around a week, made a tea, and then gave that tea to the mosquito larvae. And we did filter this infusion to get rid of any microbes that might have come from the native leaves as well. And we did the same for leaves from an invasive tree species, strawberry guava. And we also had a pure water control, which is just sterile water. And we also gave these mosquitoes 9 common bacterial genera, common to local mosquitoes around here. And we sequenced the water throughout the entire experiment to sort of understand what's happening in these early life stages. And we also collected the adult mosquitoes after they completely developed and sequenced their microbiome as well. So these are some of the results. They're kind of a lot, so I'll just go through it. This is showing all 9 bacteria genera that we gave that ended up being found in both the mosquitoes and the water. So the mosquitoes are that upside down triangle. The water is that more circular shape. And on the x-axis, we have the infusions that I was talking about, the native, the invasive, and the pure water. And on the y-axis, we have a statistical model that says if we give them these treatments, what is the probability of detecting that microbe based on the sequencing reads that I gave this program? So to just zoom in your attention here to the 2 most important microbes. So out of all the microbes that we gave them, 2 of them ended up being more enriched in the mosquitoes than the water that they came from. So if this is a simple example of the relative abundance of the Cryptobacterium in the water, the mosquito adults that came out of that treatment were much higher. So, and the same was found for a different strain of bacteria in the pure water control. So one reason this might be happening, which is what we suspect is happening, is that these microbes might be enriched because they're somehow helping the host mosquito develop. That's why they might be enriched when we sequence the adult mosquitoes. So to test that, we did a smaller follow-up experiment where we gave them, again, a whole pool of microbes. We reduced the infusion treatments down to that invasive and the pure water, where they were more abundant in the previous slide. And we manipulated whether we gave them those 2 bacterial strains, so one being the Cryptobacterium and one being the Pseudomonas. And on the y-axis here, I have what percentage of the larvae that came in emerged as adults. So in that strawberry guava treatment, that invasive leaf infusion, we saw that adding Cryptobacterium did increase the amount of adults that came out of that treatment. So that sort of supports our hypothesis that we saw from the last slide. In the pure water, the results were a little bit more interesting. So we expected to see this huge increase of adding the Pseudomonas to the pure water treatment, this huge increase in how many mosquitoes developed. But then there was this interesting microbe-microbe interaction where including both of the microbes together actually brought the overall development to look down more like what the Chrysobacterium could do on its own without the Pseudomonas. So Pseudomonas likes to be by itself for some reason in this treatment, and it does good things to help mosquitoes emerge as adults. So to summarize everything I was just showing you, we found these two strains that are probably important for mosquito development based on what we saw. However, the importance for the development really depends on the environmental context. So this is really important because most of the science going into how bacteria affect mosquitoes is happening in a lab with that sterile water treatment, right? So we might be overlooking a whole diversity of microbes that are important in other environmental contexts. And if you take these lab-reared mosquitoes, put them on the landscape for population control reasons, maybe things are not going to go the way you expect because, yeah, these microbes are different in different contexts. So with that, thank you so much to the land that this work was done on, and the HPC, and my labs, and the funders, and this is the paper for anyone that's interested, and I will try to answer any questions in the meantime. Yes.
In the first picture you showed, there was like a lighter color water and like a darker color water.
Yes.
Those different things.
You're paying very much attention. So this study, we actually manipulated a whole bunch of environmental conditions. So we did the leaf infusion, which is what I focused on for the purposes of the lightning talk, but we actually also manipulated temperature as well. So I should have maybe like cropped that figure a little bit better, but that's why we had the lighter color for a cooler treatment, and then we had the darker color for a warmer treatment. But the main factor, we also did a third treatment where we like dispersed all the water across all the mesocosms again after a few days, or like every 2 days. But we found that the leaf infusion was the biggest determinant of the microbiome composition. So we sort of like ignored the other infusions for the follow-up experiment. Nicole.
To have a captive audience, Nicola, if you could make one change to the Zumaiki gateway.
Oh, amazing. Right. So I'm a very direct, blunt person. The like version 1.0 has some bugs for some reason, and the 0.02 is the one that I used on this study, and like I can attest that it works extremely well. I don't know what's up with the new version, but yeah, someone should get on that, please. Please. We have research that depends on it. Okay.
Well, I guess if those are the end of the questions, unfortunately, you didn't get an intro. So we'll give you an outro. So Nicola is a postdoctoral researcher at PBRC, advised by Dr. Matthew Medeiros and Nicole Hinson. She earned her PhD from the Davies Lab at Boston University, where she investigated microorganisms within coral hosts, or how they are structured by reef environments. And so she went from Boston to Hawaii, to where she was in Boston studying coral, to Hawaii where she's studying mosquitoes. So that's a little bit hard to explain, perhaps, other than she gets to be in Hawaii. And also, thanks for trying to understand mosquitoes, our most the nastiest insect, I guess, most deadly insect.
Yeah, exactly. I can explain why that happened. It's because bacteria are really hard to study in corals and really easy to study in mosquitoes.
Okay. All right. Well, with that, we will move on to our next speaker, Komal Yadav. Komal comes to us from, she has a master's degree from Central University of Punjab, India, followed by a PhD in chemistry from Mysore, also in India, and joined Raysun's group here at UHM Chemistry in January 2023. And she studies current research uncovering the mechanistic pathways of chemical reactions in condensed phase. So her title is Elucidating Mechanisms of Chemical Reactions in Condensed Phase Using Computational Techniques. Thanks.
So, good afternoon, everyone. I am Komal, and I have been working as a postdoctoral research associate with Professor A. Sun for the past 1.5 years. And today, I'll be talking about elucidating the mechanisms of chemical reactions in condensed phase using some computational techniques. So, whenever we talk about reactions, we have, some reactants giving products, say A plus B, giving C plus D. And, since high school, we have been looking at this profile, I believe, where you have a reactant, and most of the time you need certain amount of energy so that you can get the products. Okay, but in reality, the curve is not that simple, and you usually have a 3n-6 dimensional surface. So if you look at the surface carefully, if you want to go from one point to another point, you have a dashed line, and that is what the profile means. So when you're doing reactions experimentally, if you want to overcome the barrier, what you do is you supply some heat to the system so that you can get the products, or you shine light on the system, or you apply some external force on the system. That is how you overcome the barrier to get products from reactants. But we are not experimentalists, and we do study reactions computationally. So the question is that how do we study reactions computationally? So there are two methods. Number one is that we study the energetics of the system. So what we do is we solve the time-independent Schrodinger's equation, and you get the energies for the reactants, transition states, products, intermediates, and so on. And then you can say that why one product is being formed and the other is not. Okay, and the other scenario is where you study the dynamics of the system. That is, you solve the Newton's equation of motion, and you see how a system evolves with time and how different products are being formed. So for the past 1.5 years, I have been studying two different reactions in condensed phase, and I'll talk briefly about them. The first one is the decomposition of FOX7. So this is the FOX7 molecule, as we can see, and FOX7 is important because it has very high detonation power. So if you want to use that as a detonating agent, you need to know how the different products are formed, why certain product is being formed, so that you can condition the reaction. Okay, so the reaction conditioning has been done already by our experimental collaborators, which is Professor Ralph Kaiser and his group. They shine light on the system and they get different products. And our job was to study why one product is being favored over the other, so that we can model the reaction. So what we do is in a simple way, we have our reactant and we have different products, and we model the reaction pathways using our chemical intuition that, which one has a higher barrier, which one has a lower barrier, why do you get one product and why don't you get the other one. So these are two simple animations that I'll be showing here, where I'm not sure why it is so slow, but you can see that, the proton is being transferred in the first one, and in the second one, again, the proton is being grabbed by a hydroxyl radical so as to form the water molecule. But this is a gas phase mechanism. And when you study reactions experimentally, you don't have a single molecule, you have a sheet. So our job was to see why, how the condensed phase is going to change the reaction mechanisms. So this is the condensed phase system, as we can see. And when you shine light on the system, the reaction can either happen from the surface or it can happen from the bulk. Now, which one is favored over the other? So as you can see, if one of the molecules is going to react, the other neighboring molecules will have to readjust, and they will play an important role in the reaction mechanism. So that is what we wanted to see here as compared to gas phase. So what we did was we studied the different pathways and the reaction mechanisms at the bulk and at the surface of the reaction. So the reaction can either happen at the surface or it can happen at the bulk. And then we try to answer which one is going to, which one is favored over the other. And briefly speaking, what we saw was the decomposition of FOX7 actually happens at the surface of the film as compared to the bulk. And this paper was published recently, earlier this year. So this was one of my projects. And the next project was to study the hypergolicity of ionic liquids. So imine CBH is an ionic liquid, and H2O2 and HNO3 are different oxidizers. Now, why this project is important? Because ionic liquids are very good propellants, and they are used as they're used as good propellants in jet sprays and in aerospace industries. So we wanted to see, when we use different oxidizers, how are the reaction mechanism changing and why one of them is being favored over the other. So again, our experimental collaborators, they found that the hydrogen peroxide, that is a very good oxidizer as compared to HNO3. And, the reaction is very, very fast when you use one over the other. So what we did was we studied the reaction computationally. So since this is an ionic liquid, what we have is we have our ionic liquid box, and you have the reactants and oxidizer, and then you try to see why one of the oxidizer is behaving very good as compared to the other. So in this scenario, what we did was we studied the time evolution of the systems. So you have two different systems. The left side one is the hydrogen peroxide system, and the right one is the HNO3 system. And you can see that in the hydrogen peroxide system, a lot of proton transfer is happening, whereas in the HNO3 system, only one of the proton is going to react. But still, why one over the other? So to answer that, we look at the free energy barriers for the reaction. And what we find is that when you look at the hydrogen peroxide system with the ionic liquid, the barrier is much less as compared to the nitric acid system. And well, that answers the question why one is being favored over the other. And yeah, so we can explain that, when you use the hydrogen peroxide with the ionic liquid, that will act as a very good propellant as compared to the nitric acid. Yeah, so these are the two things that we have been studying for the past 1.5 years. So yeah, so in the acknowledgements, I would like to thank University of Hawaii and Koa for all the computational facilities, because we don't just study one trajectory. We have like hundreds of trajectories, and that takes a lot of time. So yeah, thank you, Koa, and my PI, Professor Ray-san, and this is my group. And thank you all for listening, and I'll be happy to take any questions. Thank you.
Doing the study of those reactions in the condensed, are you treating the other molecules explicitly, or are you doing some implicit?
No,
model?
explicitly.
And then are you doing some sort of like QMMM, or do you?
We did QMMM as well as full QM.
Okay.
Yeah.
A very ignorant question. All these reactions, is this all modeling, or does this stuff happen in the lab too?
Oh, that happens in the lab too. And we try to model them to see what is because in lab you cannot, when you're doing experiments, you cannot say what is the atomic level mechanism, right? So that is what we try to figure out.
I guess if there are no more questions, thank you again. Jan Hafner. Jan, I know Jan from my past life, but currently Jan has joined the Water Resources Research Center at UH Manoa, and he's participating in the Cooperative Institute for Research Operations in Hydrology, which I guess is a project at the University of Alabama. And the objective is to evaluate the ability of several hydrological models used in operational settings to simulate stream flow over the complex terrain in Hawaii. So, and I just say that Jan, I know, has done all different kinds of modeling since I've known him quite a long time. Anyway.
Ladies and gentlemen, Ron, thank you for the introduction and for the opportunity to present some results of my new project, and it's about the hydrological modeling in Hawaii. First, I would like to acknowledge the other members of our team. Giuseppe Torre, he is the principal investigator on this particular project. Aline Falk, working with me on this project, and of course all other members of the team, as you can see, from all different departments, UH and even beyond. Our project is a part of the Cairo project, the Cooperative Institute for Research to Operations in Hydrology. And actually, it's a huge consortium having multiple agencies and universities. And we are the part, our project is a small part of this project. And now the question is why we are doing it. It's stated here. Essentially, we are trying to find, and I'm trying to find the cursor here, if it shows, is to study and evaluate abilities of hydrological models applied on extreme events in Hawaii. And the idea is if we can get the hydrological modeling right in Hawaii, which is very challenging environment for hydrology, then eventually the model or the setup would work elsewhere. So that's the overall idea or overall aim of this project. And I will mention two particular models I just got hands-on and applied to Hawaii, World of Hydro and the SUMA. So I will talk in more details later. First model, which is for quite some time in use, is called World of Hydro, and actually it is was developed as to be coupled with WRF atmospheric model that stands for Weather Research and Forecast atmospheric model. And actually it can even run standalone. And here is just the schematic of the different components. Here we have the land model with all components resulting in water balance. And once this water balance component is completed, it follows to the routing modules, which essentially redistributes the surface or subsurface water. And finally, it would result in the actual output. That's a stream flow, river stage, and flow velocities, and so forth. So this is just the overall picture how to structure this model. And what we did, we apply this model in Hawaii for selected extreme cases. One of those, I have few, but for the sake of time, I'm showing only just one. And extreme flood and rainfall event was in 21, was the 6th of December. And the maximum flow actually happened right here in backyard in Manoa Stream in the wood lawn. And this is the, in the red, this is the stream extreme event. To compare with observation, because in all those sites, there are river gauges and or stream flow gauges, and they measure. And here is the kind of metrics how we evaluate the performance of this model. There are bias, relative bias correlation, and there is one specific KGE. It is Krink-Gupta efficiency. That's the specific metrics to evaluate the match of the hydrologic model and observations. I would note, let's look more into details of those. Yeah, so as you see in each plot, you have multiple lines, and actually each line is stands for different forcing. This hydrological model is forced by the atmosphere, and we use those four forcings. The dark blue is ERA 5, that's the European model reanalysis version 5. The light blue is WRF, only atmospheric model, which is run and produced every day here locally at the Atmospheric Science Department, Dr. Chen. And then we have the two another combinations, which is the orange, I believe, and the violet. That's the ERA 5 plus MRMS. And MRMS, that's the multi-radar, multi-sensor rainfall product. That's probably the best gridded rainfall you can get the closest to the observation on 1 kilometer grid spacing, hourly frequency. And I used ERA 5, all other variables except rainfall. For the violet, you have WRF. I use all other variables except rainfall from this particular radar product. And in this plot, you see the correspondence. If you look on the blue and light blue, even it doesn't show. So it means these large scale models, even the small scale model, they don't pick up this rainfall event and it doesn't show in the model. However, using this sophisticated high resolution, high frequency precipitation product in the forcing field results in this line. Actually, it almost follows each other. So it indicates that atmospheric forcing except rainfall is not that important. So that's the one example and one of the conclusions. And in the conclusion is that rainfall is the main driving force in hydrological modeling. And you can see the comparison here. I took the difference looking on this radar product compared to the stations. This is the WRF rainfall compared to the station. And I split the difference based on rainfall intensity. And just comparing this plot, you see in for low rainfall intensity below 1 millimeter an hour, the model actually overestimates precipitation. It's blue, that means wet. However, if you go to the other side, very high intensity rainfall, you see all those colors are in red, so it means dry. So it means the WRF model, especially even the most sophisticated radar-based rainfall product, they slightly underestimates the actual precipitation. So that would be the one of the conclusions. Now the question is how to do better, and this is the still work in the progress. Is just to get and apply more sophisticated model, and this is one of them. This is the SUMA. It says for structure for unifying multiple modeling alternatives, hydrology modeling framework actually. And here it shows the complexity of this modeling framework. Each oval here represents one particular process, and actually and here you have the arrows. Actually, each process can be parameterized by different parameterization schemes. So we have about 27 parameterizations to select, and each parameterization there is also a parameter constant that describes, like for instance here, thermal conductivity can have those values as shown here. Now the question is, how do we pick the best combination? And for that we use the optimization and calibration, which essentially means you take the model, you select, or I did select some parameterization options and run it until we will get the best match with observations. That's the optimization and calibration. You select those 34, this 34 constant, 34 important, and run the model in iterative fashion to minimize the error with observations. That's where the high performing computation and Koa system comes in. Here are some preliminary results. I'm still working on how to match the actual observation. And this illustrates the difference in how sensitive the model is on selecting. Here we have the hydrological routing in time delay option. Here we are comparing it with the instantaneous runoff. Again, the red line is the observation. Here, the green line is the model. And see, the switching the routing options in the hydrological model would result in dramatic difference. And I think this on the right seems to be on right track compared to that one on the left. And with that, I would like to thank the HPC staff for the resources, and more importantly for the help with the models, with the script, with everything. And special thanks to David, because he is very patient and very helpful with all my problems. And thank you very much.
Well, I'll ask a question. How close to putting this into operation do you think you are? Either of these models? I know the point of doing the modeling is to actually get them into operation, and although you wouldn't be doing that work.
I will not be doing that. That's still, I think it's up to the NOAA.
Okay.
Kevin Kodama is part of, I think he was also mentioned on part of the team. He is in the operations. And actually, I think that's the whole overall idea is because right now, I think it is NOAA, they are running the national water model. And which it is old and it's not doing that well. That was the reason why we are trying to find other ways to implement a new model. And naturally, Hawaii was the best test bed for that, because if it works in Great Plains, that's kind of boring. You have more interesting weather and more interesting flooding events in Hawaii. It's more challenging from a modeling point of view. So to answer that question, I don't know.
That's a fair answer. That's the one I use quite a bit. Okay, well, any other questions? Not, I guess we'll plan. Oh, have one. Go ahead.
It's a simple question, but are you modeling other islands than Oahu?
Yeah, other islands also. I just picked for because otherwise I would pick you.
Yeah,
And yeah, that's still it is actually it's my I switched from climate models to hydrological models about a year ago and few months. So this is still I'm still on the learning curve, so to speak. But that's for the whole Hawaii.
And that's it.
Well, the Oahu is better because we have much more observation points for rainfall and for gauges in streams. Thank you.
I'll continue on welcoming Leandro Viscardi, also from Giuseppe Vittori's group. So Leandro is a postdoc in atmospheric sciences at UH Manoa. He received a PhD in physics from the University of Sao Paulo, where he conducted research on the environmental controls of isolated convection in the Amazon. Currently, his project named Change Hawaii is focusing primarily on the process of precipitation recycling over the Hawaiian Islands and investigating how changes in land use and land cover impact climate in Hawaii.
Thank you. So thank you for the opportunity to let me share my work in this high performance computing symposium. The title of my presentation is Modeling Rainfall Recycling over the Hawaiian Islands. To start, tropical islands are biodiversity hotspots, yet many are highly vulnerable to the impacts of climate change. However, many of these tropical islands, including Hawaii, have undergone dramatic changes over the past few decades, for example, associated with shifts in land cover. So one specific research question that arises in this context is how much of the rainfall in Hawaii originates from water evaporated from the islands, a process known as rainfall recycling, or using other words, how much the islands in Hawaii can control the rainfall occurring over the island. So how much the islands can control their own weather, which is associated with the rainfall recycling. So the objective of our research is to evaluate the rainfall recycling over the Hawaiian Islands using numerical simulations, more specifically the model called the Weather Research and Forecasting Model, WRF, version 4.3.3, as using by the previous speaker, Jim. So this model simulates the weather, the clouds, and rainfall. And you are also combined with this model, a module called the water vapor tracers, this model allows us to tag water coming from the islands, okay, so that you can quantify the fraction of rainfall occurring in Hawaii, which is associated with this water evaporated from the islands, the rainfall recycling. So our simulations are covering the period from 2015 to 2019. Now I'd like to share an animation, the model results for June 2018. Here the gray scales indicates the clouds, the cloud fields during the simulation. So it's essentially a satellite view of the clouds, and the colors indicates the rainfall recycling. Okay, so as you can see in this animation, most of the rainfall recycling occurs over the Hawaii island. Okay, and it also oscillates with time. So most of the rainfall recycling occurs during daytime hours. It peaks around noon, and then it decreases, and it has little rainfall recycling during nocturnal hours. And you can also notice from this animation that most of the rainfall recycling occurs around the regions of the mountains. So now I will share this next slide. Here I'm showing the composite results during the entire simulation. The left panel indicates the result during the dry season from May to October, and the right panel shows the results during the wet season from November to April. The colors indicates the rainfall recycling, and here I also included these arrows, which is the winds, the trade winds, the winds blowing towards the islands. It's well known in the literature that the winds in Hawaii are stronger during the dry season, as you can see here, and the interaction between these stronger winds during the dry season and the topography enhances precipitation rainfall during the dry season. And in your results, you're also showing that this also increases the rainfall recycling. Now I go to the next slide. Okay, sorry. So here, I'm showing the rainfall recycling during the simulation. However, in this figure, I'm showing the rainfall recycling as a function of the topography height, okay, surface height. Then you have results for daytime period from 10 a.m. to 6 p.m. and nocturnal hours from 10 p.m. to 6 a.m. As I mentioned earlier, it has little rainfall recycling during nocturnal hours. As you can see here, most of the rainfall recycling occurs during daytime hours, and the increase approximately linearly increases with the topography height. So basically, it has a strong correlation between the rainfall recycling and the topography. Now, I'm showing the results associated with the impact of land cover on rainfall recycling. So essentially, we have our control simulations with the Hawaii land cover. We also conducted a simulation where we replaced the land cover from Hawaii with a forest, okay, which is a condition that would increase the amount of water vapor released from the islands, the evaporated water from the islands, and then to contrast this condition, you also conducted a simulation where the land cover was replaced with parent soil, and then it's a condition with zero vegetation, and then you can see from these results that the vegetation also impacts the rainfall recycling. Okay, so with a forest, for example, you'd have an increase of rainfall recycling by up to 5% compared with the real land cover in Hawaii. So to conclude, while instantaneous rainfall recycling can reach high values greater than 80% locally, the overall mean climatology, the long-term average, remains below 10%. Hawaii Island shows the highest rainfall recycling, averaging 2% in the wet season and 3% in the dry season, and peaking at 6 and 8% over the mountains. Daytime rainfall recycling increases almost linearly with terrain elevation, and land use and land cover change may alter mean recycling by up to 5%. Thank you very much.
Questions?
The study was only from 2019 or 18. I was curious if you thought your first bullet point on the conclusion slide, do you think that's going to change at all with global climate change, or do you feel like it's going to be a similar kind of peaks with a generally low peak, or do you see that changing at all?
I didn't understand the beginning of the question.
Oh, I was just curious if you think that first point will maybe change at all with kind of larger global weather patterns shifting, or do you think that it'll probably stay the same in Hawaii?
The rainfall recycling.
Yeah, it's kind of, or if you go back to the last slide you were on. The one with the points, your conclusions.
Okay, the points, which point?
Yeah, just the first bullet point.
Okay.
I was just curious if you thought that would shift or change over time at all.
If it changes this climatology with time in Hawaii or another region?
In Hawaii.
In Hawaii. So the second line I'm showing here, it changes with the season, the rainfall recycling. The precipitation patterns also depends on the season, but it's small the difference. So in Hawaii, the rainfall recycling, it's about 2 to 3% on average over the Big Island, and for other islands, the rainfall recycling is smaller, for example, so it depends on the island size, the vegetation, and then if you change the vegetation, the island size, or if you go to a region with forest, like the Amazon forest, the rainfall recycling, it's about 25%. So that will change. But over Hawaii, it's very much dependent on the island.
Well, have no more questions. Let's thank Leandro one more time and all our speakers, and thank you.